Well, I think it's time for our webinar to begin. And I would like to introduce myself. I am Kathy Grady. I chair the ISHLT Grants and Awards Committee. We also have expert judges who will be judging this session from both, they are from both ISHLT and Enduring Hearts. And with that, I would like to welcome everyone to the ISHLT Enduring Hearts Pediatric Heart Transplant Innovation Challenge Grant Finalist Presentation Session today. We have 5 presenters, amongst whom a finalist will be selected who will be awarded this challenge grant. And I wish everyone the best of luck. I would like to remind our presenters that they will have 10 minutes for their presentation, followed by 3 to 4 minutes for questions. I ask all attendees, if you have a question, please put your question in the chat and I will read that question out loud to the presenter. So with that, I'd like to begin with our first presentation. The presentation is entitled, The Impact of Adverse Childhood Events on Cardiometabolic Risk in Pediatric Heart Transplant Recipients, presented by Carmel Bogle from the Johns Hopkins Children's Center, Baltimore, Maryland. Thank you.
Thank you so much for the opportunity to present. Oops. Make sure my video is on. I'd like to thank ISHLT and Enduring Hearts for the opportunity to present this work of research. My name is Dr. Bogle and I'm at Johns Hopkins. And as stated, I'd like to present the impact of adverse childhood experiences on cardiometabolic risk factors in pediatric heart transplant recipients. So adverse childhood experiences, or ACEs for short, describe various stressors that include household dysfunction, environmental stressors, or abuse that an individual can experience during childhood. The impact of ACEs has been noted to have long-lasting effects on individuals that reach well into adulthood. This impact has been shown to have significant impact on cardiometabolic risk factors in the general population, including obesity, hypertension, diabetes, and dyslipidemia. It is also noted that the number of ACEs is correlated to an increased number of inflammatory markers, which has been projected to influence these cardiometabolic risk factors and cardiovascular disease. In regard to our transplant world, we know that these cardiometabolic risk factors have an effect on the long-term outcomes of our transplant recipients. To discuss our tool that we will be using the PEARLS assessment tool, which stands for the Pediatric ACEs and Related Life Event Screener, which has been validated in multiple pediatric population studies. This tool has translations in up to 18 languages, with patient assent available for patients that are 12 years and above. There's a score of 0 to 10, and as you can see on this slide, is a complete survey, which takes 10 minutes or less to complete. It has a score of 0 being low risk, medium being 1 to 3 risk, and high risk being 4 or greater. In addition to the cumulative score, based on this assessment tool, one would be able to differentiate between interpersonal and environmental ACEs. Why we believe this is significant is, in a previous Enduring Heart study, it was noted that cardiometabolic risk factors play an impact in our pediatric heart transplant recipients. On this graph, we can see the freedom from CAV and years after transplant, with the yellow line being obesity, in which we can see that the freedom from, there's an increase of CAV related to our obese patients, as well as graft loss that we see on this next slide. Additionally, the study found that recipients of minority status carried an increased risk of graft loss compared to their white counterparts. With research focusing on ACEs, it is noted that there is a correlation between number of ACEs and cardiometabolic risk factors. On this slide, on the y-axis, we see the number of members with the condition within the clusters, with the 2 highlighted in the middle being CRP as a marker for inflammation and metabolic risk factors on the 3rd panel, with the increased dark lines representing increased childhood experiences. So as we can see, there's an increase of CRP as a marker for inflammation, as well as cardiometabolic risk factors. Delving deeper into ACEs and cardiometabolic risk factors, those that have high ACE scores have more than a 2-fold risk of cardiovascular disease, as well as more than a 2-fold risk of premature death in adults, which can be seen here on the solid line, in which the solid blue line represents those that have had a high ACE score of 4 or greater, with a significantly less survival period. The prevalence of ACEs in the US has been found to be disproportionate among racial minorities, children with special health care needs, and those that live in rural regions, as well as those with lower socioeconomic status. Although all races respond to ACEs, although all races respond to ACEs in similar ways, there is an increased prevalence within racial minorities. Multiple studies in the pediatric as well as adult heart transplant population have noted that minority recipients are at higher risk for CAV, recurrent rejection, and need for retransplantation. It's important to note that these racial disparities are noted even when controlling for social economic status. What our study's long-term goal is to determine the impact ACEs have on risk factors that can impact the long-term outcomes in our pediatric heart transplant recipients, with our central hypothesis being that we believe the number of ACEs has a direct relationship to increased metabolic risk factors, as well as inflammation, which can impact the development of CAV, frequency of rejection, and overall survival in our pediatric heart transplant recipients. We believe that this research practice is in alignment with the grant, as it evaluates the risk factors that lead to CAV and rejection through the innovative approach of evaluating the impact of ACEs that it can have on cardiometabolic health. This approach integrates how psychosocial factors play a role in the overall health of our pediatric heart transplant recipients, in which our minority recipients appear to be at increased risk for prevalence of ACEs. We believe that a better understanding of the impact of ACEs could overall lead to more interventions and perhaps policy changes to overall improve graft survival. Our primary aim is to determine the association between ACEs and BMI percentile category, with the BMI percentile category being a marker for cardiometabolic risk factors, with our hypothesis being that there would be an association between the number of ACEs and BMI percentile. Our secondary and exploratory aims are to examine the association between ACEs and additional cardiometabolic risk factors, such as a lipid panel and hypertension diagnosis, as well as exploring the association between ACEs with time to event for analysis for transplant events, such as CAV, rejection, the need for retransplantation, and hospitalization within the first year of transplant. In regard to the BMI, we will be using the category of overweight and obese based on the CDC criteria as can be seen. And for our lipid category, we'll be using ideal, intermediate, and poor based on the National Cholesterol Education Program criteria. Our methods include a cross-sectional pilot study to evaluate the impact ACEs have in the pediatric heart transplant population. ACEs will be collected using the PEARLS assessment tool as reviewed during routine heart transplant clinical visits for patients that have survived at least 1 year post-transplant and are 6 years and above. Clinical data will be collected at this time with the most recent clinical data, and we expect that the data will be collected over a 6-month period using at least these 4 participating Pediatric Heart Transplant Society centers. For our primary outcome analysis, we will be focusing on BMI, in which we will be using categorical variables expressed as percentage and comparing using chi-squared analysis or Fisher exact test as appropriate, as well as chi-squared analysis and logistical regression for overweight and obese category for those that have a Pearl score greater than or equal to 4. Our secondary and exploratory analysis will be focusing on the lipid panel, hypertension, and time to event complications, in which we will evaluate low and high risk Pearl score as categorical variables, controlling for risk factors using multivariable logistic regression, survival analysis utilizing Kaplan-Meier method, and the log rank for time to event for heart transplant complications. We recognize that our study may have some limitations and pitfalls, and that our high-risk patients might not even be suitable or listed as a heart transplant candidate, or furthermore, that these high-risk patients may not survive to 1 year post-transplant. It is important to note that our PHTS centers that are involved have an average of 95% or greater for survival for 1 year post-transplant. Therefore, we believe the number of patients that would be lost is relatively small. Also, as inherent in any survey, ACEs may be underreported as it is dependent on the participants to answer truthfully. Also, something that may result from ACEs screening is that there might be unexpected follow-up or resources that these patients might need. But as our centers are PHTS centers, we know that we will have the support needed to support these families during any unexpected follow-up or resources. We believe this study is important as this will be the first study to evaluate the association of ACEs and cardiometabolic risk factors within the pediatric heart transplant population. This study will be able to further extrapolate whether interpersonal or environmental ACEs have a different impact on the cardiometabolic risk factor. And furthermore, this study will explore the impact ACEs may have on pediatric heart transplant recipients' long-term outcome, including the development of CAV and the number of rejection. To discuss our study team, I have a special interest in cardiometabolic risk factors and its overall impact in the pediatric population. Dr. Auerbach is also a pediatric heart transplant physician and medical director of the VAD program at Children's Hospital, Colorado, and current Pediatric Heart Transplant Society president. He has a special interest in the predictors of outcomes in pediatric heart failure and transplantation. Dr. Brosby is an assistant professor and psychologist that works with our pediatric heart transplant population, with a special interest in childhood traumatic stress, and is co-founder of the Medical Traumatic Stress Special Interest Group of the Society of Pediatric Psychology. Dr. Van Eck is an assistant professor of psychiatry and behavioral sciences, with a special interest in screening programs for traumatic exposure and mental health as a tool for engaging our at-risk youth. Dr. Casino is an associate professor and director of psychosocial services and research for the University of Michigan Congenital Heart Center. She is a world expert for the impact of psychosocial outcomes in pediatric advanced heart disease. I'd like to thank you so much for your time and welcome any questions.
Well, thank you so much, Dr. Bogle. We have two questions that have been posted. The first question, how will you separate lack of compliance due to ACEs from role of obesity?
I think that's a good question. I think for lack of compliance versus ACEs, I don't know if we can extrapolate that with this pilot data at first. We can include that as a diagnosis within the clinical research, but I think our primary focus will be focusing on cardiometabolic risk factors and how it relates to ACEs. We'll use a multivariable regression to see if we can filter that out with a statistical analysis.
Thank you. And we have a second question. You seem relatively convinced from the data presented that ACEs will be related to risk factors and presumably therefore outcomes. If your research goes on to prove this hypothesis, how will it change our management? Shouldn't we be doing everything to reduce ACEs regardless?
That's a great question. So I think if we do find that there is an actual connection, I think this could lead to perhaps more policy change, as well as more on a national as well as hospital-driven policy change. And if we know that certain patients are at more high risk and have a higher ACEs score, that might lead us to determine, do these patients need higher follow-up? Do we need to monitor their immunosuppression regimen a little bit more so that we can address it before it becomes an issue where we're several months or several years down the line of having non-compliance? That's a great question.
Thank you very much. Third question. The differences in the registry studies you showed appear to be quite small. How likely is it that you will be able to identify unique differences in a smaller cohort, and how will you control for the factors you showed associated with both poor outcomes and ACEs?
So for our power calculation, we suggested for a Pearl score of greater than or equal to 4, which would be the high risk category, and those that would be overweight and obese, that we would need at least 66 patients to determine if that is significant or not. And so I think from our high volume centers that we're pulling from, we'll be able to reach that even with a 70% attrition. And I think from the multivariable analysis, we'll hopefully be able to filter out some of those risk factors, including asking providers about risk of compliance or not compliance. So that is obviously can be gray and is dependent on the provider. So it does have some risk factors and limitations as discussed, but I think it would be the first study that has even been explored within the pediatric population. So I think it'd be important data for us to use. And then our plan would be to potentially use that for a more longitudinal study to explore those other risk factors that we might not even be able to delineate within the pilot study itself.
Thank you. Another question, what is the projected sample size?
So we would need for our power calculation at least 66 patients. So we would hope to have at least greater than that. I would expect that we would have at least 80 to 100 patients, and are actively continuing to recruit PHTS members, especially if we were to do a secondary study. But we would need at least 66 that we would expect to show any type of power calculation for significance.
Okay, thank you. I also have a question regarding use of your PEARLS tool. How are you going to prevent missing data? And if there is missing data, how are you going to handle it?
That's a great question. I think in general, if there's more than 80% of missing data from that survey tool, then we would have to not use it or exempt that from our study. I think if it is missing, if it's within that time frame, then I think we would probably have to note that within our study process and help determine the environmental or interpersonal to see if there's any trend, if there's any particular question that's being missed, and help to address that in further analysis.
Okay. Thank you very much. I think it is time to move on. We very much appreciate your presentation. Thank you. Okay, so our next presenter is presenting on clonal hematopoiesis in heart transplant recipients. This is presented by Lakshmi Gokanapudi Han from Washington University in St. Louis, Missouri. Thank you, Dr. Han.
Thank you. Looking forward to talking to you guys about clonal hematopoiesis in heart transplant recipients. My research mentor is Corey Levine, who is an associate professor at WashU. He is another heart transplant physician, but for adults. Over the past several decades, advances in medical, surgical, and device-based therapies have improved outcomes for heart failure patients, but many patients still progress to advanced heart failure, and heart transplant is a transformative, or can be a transformative and life-saving therapy for these patients. The success is dictated by donor organ availability, immunological compatibility between the donor and recipient, and the ability to tolerate systemic immunosuppression. Major morbidity and mortality happens due to primary graft dysfunction, heart transplant rejection, and complications of systemic immunosuppression, including infection and cancer. There's still a paucity of information pertaining to modifiable risk factors and targetable mechanisms that contribute to these adverse and life-threatening complications. Clonal hematopoiesis is a phenomenon that's found in healthy older individuals, as well as children and younger adults with chronic diseases. It occurs when hematopoietic progenitor cells acquire mutations that confer a fitness advantage relative to the background population, and these mutant progenitor cells then expand, giving rise to a substantial portion of peripheral blood cells. Whole exome sequencing has shown that variants in genes such as DNMT3A, TET2, they're the most frequent causes of clonal hematopoiesis. And mutations, particularly in those two genes, have been shown to have the strongest association with cardiovascular mortality and are prevalent in heart failure populations. So while this has been studied in heart failure populations and widely described to be deleterious, the effects of clonal hematopoiesis on solid organ transplant patients, including heart transplant, have yet to be explored. So to gain initial insights into the impact of clonal hematopoiesis on post-transplant outcomes, we used a mouse model of allogeneic heart transplantation. And surprisingly, we found that recipient clonal hematopoiesis reduced allograft rejection and promoted long-term allograft acceptance, indicating that clonal hematopoiesis in the transplant population may confer protection instead of being deleterious. So my goal is to study clonal hematopoiesis in adult and pediatric heart transplant recipients to define the clinical relevance of clonal hematopoiesis on heart transplant outcomes, including rejection and allograft failure, and to gain mechanistic insights by employing our expertise in single-cell technologies to address how clonal hematopoiesis alters the transcriptomic and epigenomic landscape of peripheral immune cells before and after transplantation. This project will use our actively enrolling heart failure registry that is at WashU, and also our next generation sequencing pipeline that is designed to detect clonal hematopoiesis. In terms of innovation, the conceptual innovation would be identifying a mechanism by which clonal hematopoiesis promotes allograft longevity and decreases the incidence of rejection. And this has the potential to impact recipient selection as well as influence post-transplant immunosuppression strategies. The technical innovation is that in collaboration with our Genome Institute, we developed a panel containing 246 genes that are commonly mutated in clonal hematopoiesis and myeloid malignancies. A second technical innovation of our project is to utilize our expertise in single-cell RNA ATAC sequencing, which we'll talk about more in a little bit, to investigate whether CH driver mutations impact gene expression and chromatin accessibility at single-cell resolution. To evaluate the clinical relevance of the observations from our mice studies, we established a pediatric and adult transplant registry at WashU and measured clonal hematopoiesis in 40 pediatric and 120 adult advanced heart failure patients as a pilot study. We found that clonal hematopoiesis was detected in 20% of our patients, and as expected, it was more prevalent in the older age groups, with some patients having multiple mutations. The most frequently mutated genes were similar to what we find in the literature, which was DNMT3A and TET2. Again, surprisingly, instead of being deleterious, we found that clonal hematopoiesis was associated with allograft longevity in our patient population. So among patients who underwent heart transplantation, the composite outcome of cardiovascular mortality greater than 2R cellular rejection, antibody-mediated rejection, and primary graft dysfunction was significantly reduced in patients who had clonal hematopoiesis with a p-value less than 0.018. Again, these patients with clonal hematopoiesis, which is the red line in this graph, survived longer than those without clonal hematopoiesis with lesser number of rejection episodes. To build upon our pilot study, this proposal hopes to test the hypothesis that clonal hematopoiesis is associated with reduced incidence of donor heart rejection and failure, and that it alters the transcriptional landscape of peripheral immune cells, particularly T cells and monocytes, and leads to immune tolerance or graft tolerance. So the first aim is to increase the post-transplant clinical cohort to validate our pilot study, which looked at the impact of clonal hematopoiesis on post-heart transplant outcomes, such as greater than 2R cellular rejection, antibody-mediated rejection, primary graft dysfunction. In order to do this, we'll use our next-generation sequencing pipeline and sequence the next cohort of adult and pediatric patients and look at associations between clinical demographics, clonal hematopoiesis, variant allelic fraction, and perform a competing risk analysis to determine the key variables that are associated with improved survival and decreased rejection. We established pediatric and adult advanced heart failure registries with comprehensive clinical phenotyping, and we continue to enroll into those registries. Our inclusion criteria is all pediatric and adult patients undergoing evaluation for heart transplant or mechanical support, and we exclude any patients who are actively infected, have HIV and hepatitis. We collect blood samples from these patients, which are then processed into plasma and peripheral blood monocytes that are cryopreserved for DNA sequencing, flow cytometry, and then the rest of our functional studies and our single cell studies. CH is measured using our custom next-generation sequencing pipeline, and we use a variant allelic fraction cutoff greater than or equal to 2%. We plan to validate our pilot study by genotyping an additional 70 patients, and this should provide sufficient statistical power to identify a relative hazard ratio of 2 using a two-tailed p-value less than 0.05. This is just a general schematic of how our next generation DNA sequencing works. We collect the blood, we process it, and from the Buffy code, we obtain the PBMCs, we extract the DNA, we prepare our libraries using our customized clonal hematopoiesis gene panel, and then do the DNA sequencing and the analysis. So we use next generation DNA sequencing, which is also known as gene panel sequencing, because it's more sensitive than standard exome sequencing, and that's why we prefer that. We plan to validate like we talked about, our pilot study by genotyping an additional 70% patients. And we'll look at the associations between clonal hematopoiesis, using it as both using it as a dichotomous variable. So the incidence of clonal hematopoiesis continues, and also as a continuous variable incidence with variant allelic fraction, and clinical demographics, and perform a competing risk analysis to test the hypothesis that clonal hematopoiesis is associated with improved survival and reduced rejection. Anticipated results, we hope to find that at least 20% of our patients will have clonal hematopoiesis, and we also hope to validate the frequency of clonal hematopoiesis in our patient population, the most prevalent clonal hematopoiesis driver genes, and the clonal abundance based on measurement of VAF. And we hope to validate our pilot study and see that clonal hematopoiesis in heart transplant recipients is indeed associated with reduced incidence of donor heart rejection and failure. In terms of aim 2, this aim is to identify the mechanistic basis by which clonal hematopoiesis may confer allograft protection. So we'll leverage our expertise in single-cell technologies, single-cell RNA assay for transposase accessible chromatin sequencing, also known as scRNA ATAC sequencing, to address how clonal hematopoiesis changes the transcriptomic and epigenomic landscape of the peripheral immune cells before and after heart transplantation. These studies will provide insights into the cell types impacted by clonal hematopoiesis and how that has resultant effects on transcriptional network activity, and that'll give us information regarding the mechanisms by which clonal hematopoiesis impacts the immune system of heart transplant recipients. scRNA ATAC sequencing. So the many genes, clonal hematopoiesis genes, especially TET2, DNMT3A, regulate gene expression through effects on chromatin accessibility. So the scRNA ATAC sequencing is a powerful method to investigate whether CH driver mutations impact gene expression and chromatin accessibility at single cell resolution. So this gives us a unified view of a cell's open chromatin landscape and their gene expression profile, and you can resolve how cell types and states are established, find gene regulatory interactions, and interpret epigenetic profiles with key expression markers. And as an example from my lab, I've included an image of how you can analyze how the cells are clustered and how you can see differential chromatin accessibility or gene expression. So we hope to perform scRNA ATAC sequencing before and 3 months after transplant on the following sex and age matched cohorts. So 10 subjects without clonal hematopoiesis, 10 with TET2, and 10 with DNMT3A clonal hematopoiesis. And we hope to use 10 samples per group so that we can use pseudo bulk analysis and provide greater statistical power, allowing us to deal with interpatient variability. These studies, again, will be performed on cryogenic ABMCs. Our anticipated results are that clonal hematopoiesis alters the transcriptomic and epigenomic landscape and decreases the ability of peripheral immune cells to present antigens, leading to prolonged allograft tolerance. Impact, we hope to establish clonal hematopoiesis as a new modifiable risk factor for heart transplantation and open up a new direction for the heart transplant field by allowing us to develop personalized immunosuppression regimens. Thank you so much, and I can take any questions.
Okay. Well, thank you so much, Dr. Hahn. We have questions that are coming in, so let's get started. First question, what mechanisms by which do you hypothesize that clonal hematopoiesis confers a survival advantage?
Yes, so we think that it somehow alters how antigens are presented. So we think that there will be a change in how T cells and monocytes look at the graft as a foreign antigen.
Thank you. Okay, next question. How many pediatric patients will be in your cohort if only a small percentage of these have CH, and given that they will be a heterogeneous group with regard to age at the least, are you convinced that you will be able to draw strong conclusion from the pediatric group?
Yeah, that's a great question. So, sorry, let me go to Oh, I guess I didn't include that slide. So far we've sequenced 40 pediatric patients, and among the 70 that we plan to sequence, we have at least, I believe, 25 that are pediatric patients. Of those, we have, so the group that's already been sequenced, we have about 20% of pediatric patients who have clonal hematopoiesis, which is actually comparable to the population. So I think that we will be able to at least draw some conclusions based on both the pediatric and the adult data.
Thank you. Next question. Do you have a hypothesis as to why there are differences in pediatric to adult recipients to CH? And any thoughts on the effects of age on CH?
Yeah, so we actually noticed, based on our initial pilot, that the variant allelic fraction of clonal hematopoiesis did not change and was not significantly different, regardless of whether the patient was pediatric or adult. So I believe that whatever effects clonal hematopoiesis has on the immune system when it is present, it's the variant allelic fraction does not change. So I think that whatever effects we see will be comparable in both pediatric and adult population. It is true that it's more prevalent in the adults versus peds, but I think what we're finding is that there's enough peds patients with clonal hematopoiesis. So we should be able to see a similar difference. But I'm looking forward to it when we do this study to see what mechanistic basis we find and whether there's any different cell clustering that happens between adult and peds population.
Thank you. And last question. To which extent are the changes in CH result of clinical management factors, time post-transplant, type and intensity of immune suppression, age, previous thymus excision in congenital heart disease? Is this reflecting a primary factor or just indicating more intense immune suppression?
Yeah, so based on what we're finding, it's surprising, first of all, that instead of being deleterious, we're finding a survival advantage, so to speak. And our population so far is sex and age matched and all of that, adult and peds. It's true, it's a multifactorial thing leading to survival. But what we hope to actually do is, if patients with clonal hematopoiesis have a survival advantage and they, like if there's an issue or the mechanism is that they have less propensity to detect the graft as a foreign antigen, then these are patients who could potentially benefit from less immunosuppression. And so we will be able to actually change how we manage these patients by, if we know this right off the bat, whether they have clonal hematopoiesis or not, we should be able to hopefully put them on lower immunosuppression regimens and decrease their risk for, the immunosuppression complications such as PTLD and things like that.
And one final question actually came in. It's a quick question. Based on ethics restrictions of blood collection to 1 milliliter per kilogram, can you run your test with smaller sample sizes, or do you need to exclude smaller children?
No, actually, some of our pediatric patients are infants who are a few weeks old when, especially if they're undergoing VAD placement, that becomes the first time that we collect blood for the registry at that point. And we've not had an issue. We can run our tests on pretty significantly small amount of blood volume.
Okay. Thank you so much, Dr. Hahn, and we will move on to our next presentation. And that presentation is establishing the feasibility of native CMR imaging for the diagnosis of cardiac allograft rejection in pediatric orthotopic heart transplant recipients. The finalist is Dr. Peter McDonald from St. Vincent's Hospital in Sydney, Australia. And this proposal is presented by Andrew Jabbour, also from St. Vincent and Sydney, Australia. Dr. DeBoer.
Thank you very much, Kathy. Can you hear me okay?
Yes, we can.
I
And
might
we
just-
see your presentation.
Fantastic. So I'm just going to try for one more. I think I'm just going to try my video just once more because I've made a permissions change on my laptop, but I can't start it because I think the host needs to give me permission to do so. Let me see if that works. Okay, and my apologies for the video, but you might hear some Kookaburra bird song, which will prove that I'm here in Australia presenting this talk. So thank you for your time, and thank you for the opportunity to present some of our research and our grant application. So we're looking at establishing the feasibility of native CMR for the diagnosis of cardiac allograft rejection in pediatric orthotropic heart transplant recipients. It comes as no surprise to this audience that the first year after transplantation, there's substantial attrition. And this mostly comes from rejection, graft failure, and infection in the first year. And it's really a balance in that first year of not giving too much immunosuppression so that we cause substantial infection, but giving enough so that we don't cause so that we can prevent rejection. And we've done this for decades by a technique called endomyocardial biopsies. And we use endomyocardial biopsies. We look at the samples under a microscope. We see lots of lymphocytes. We can look at the cell structure, and we can grade, according to ISHLT criteria, the degree of rejection or immune infiltration within the myocardium, and then treat accordingly. It's not perfect, but it has served us pretty well for decades. There's also endomyocardial biopsy also allows us to assess irreversible injury with graft failure. But even though biopsies really have saved so many lives over the last 5 decades, they don't come for free. They're invasive. Patients don't like them. There's a very small proportion of patients that unfortunately suffer serious complications from the biopsies. They're expensive and they're inconvenient. Perhaps there is a better way to do this, and we believe that there is. Cardiovascular magnetic resonance can detect edema, and we know that edema goes with inflammation and rejection. Using T1 and T2 weighted relaxation curves, you can acquire images that can quite accurately detect myocardial edema. And this has been demonstrated not just in heart transplantation by our group and many others, but also it's been demonstrated in the non-transplant population in the detection of myocarditis and in the recovery from myocarditis, viral myocarditis, etc., after treatment with immunosuppression. So we've had a program of looking at heart transplant rejection using CMR over the last decade, and we've published this data in high-ranking US journals, amongst others around the world. This paper here on the left was our sentinel paper that looked at T1 mapping in the diagnosis of cardiac allograft rejection, and I'll go through some of that data shortly. And this year, we published a prospective randomized study that assessed the role of CMR in cardiac transplantation. And I'll go through that shortly as a background to what we want to now do in children. So you can see that, I'll just move. So you can see here in our validation work that we looked at non-heart transplant healthy controls, those with heart transplantation that had a contemporaneous biopsy within 24 hours, and those that had no rejection, mild rejection, 2R rejection, and those that had antibody-mediated rejection. And whether you looked at T1 mapping, sorry for the typo, T2 mapping, whether it was T1 or T2 mapping, you can see that with more rejection, you get higher values on both T1 and T2 mapping. And it's quite sensitive. It's very, very sensitive. And the strength of this technique is to rule out rejection in those patients who are undergoing screening biopsies, because we should always remember that the majority of screening biopsies are either normal or demonstrate mild rejection, and then the alteration to the immunosuppression regime thereafter is minimal. You really want to pick those who've got significant rejection. And perhaps there's a better way, rather than grading them into 3 classes, perhaps there should be a continuum of rejection where we can individually tailor immunosuppression. So we have an excellent negative predictive value using T1 and T2 mapping of nearly 99%. So we took this data and then we randomized 40 patients to either CMR or endomyocardial biopsies only. And then we followed them for a year. Though at any stage, anyone in the CMR group could undergo a biopsy if the clinician was worried that the CMR just wasn't right. Likewise, anyone in the endomyocardial biopsy group could have a CMR if, for example, they had access issues or other, although that occurred extremely rarely. This mosaic plot is quite an important plot. So I might draw your attention to this top plot. This is the endomyocardial biopsy group. The bottom plot here is the CMR group. It's color-coded into no rejection, 1R rejection, and significant 2R rejection. In the endomyocardial biopsy group, each color, each dot on this plot represents a sampling time point. And you can see here that each horizontal line represents an individual within the study. And you can see here that this individual had 1, 2, 3 2R rejections. This had 2, 1, 1, 1, 1, 1. And then these patients didn't have any substantial 2R rejection. They just had 1R rejection. So this is the CMR biopsy group. So the CMR group, and in the CMR group, you can see in this group that the number of substantial rejection episodes. The total number of patients in the CMR group and the total number of patients in the endomyocardial biopsy group that had significant rejection were the same. The total number of endomyocardial biopsies that showed more significant rejection was slightly higher, although not statistically so. Each little circle here represented a time point in the study where the clinician was worried that the MRI may not be accurate. And so the patient then underwent an endomyocardial biopsy to be sure. And on each of these occasions where you see a circle, the MRI was spot on. It was absolutely spot on. Where you see a triangle, the CMR had overcalled the degree of rejection by 1. And that was only in two cases in this whole situation, in this whole group, and of all of the 11 check biopsies that the patients underwent. So the CMR is doing a great job in this second study, this prospective randomized study. If you look at freedom from rejection, there's no difference between the endomyocardial biopsy and the CMR. If you look at freedom from greater than 2R rejection, there's no difference between the CMR group and the endomyocardial biopsy group. If you look at T1 mapping values and T2 mapping values, you can see that those that had significant rejection before and after immunosuppression, and you can see their values drop after they've been treated with immunosuppression. So we really believe that this is the right way to look at rejection as a screening tool. And we've proposed in future studies that we use CMR first and then look to perform an endomyocardial biopsy only where there's doubt, as we did in this initial pilot study. If you look at the total immunosuppression exposure in the patients between the endomyocardial biopsy group and the CMR group, they were identical. The total oral prednisolone dose over the course of the year was the same. The amount of IV methylprednisolone administered to patients over the course of the year was the same. The tacrolimus levels between the groups were the same. And the total corticosteroid dose between the groups was the same. So we're not over or underexposing patients to immunosuppression. If we look at infections, in fact, we found that for some reason, and perhaps this was just a statistical anomaly, and we're going to look at this again in the next phase of study, perhaps there was less viral infection, and this led to a slightly reduced number of total hospitalizations driven by less viral-mediated hospitalizations. Something, again, we have to look at in our next phase. If you look at the freedom from hospitalisation in total, the groups do overlap quite significantly. And so we're not hospitalising patients anymore using MRI. And this is the big clincher. Within the Australian healthcare setting, you can see here that if you look at the cumulative cost of the procedures over the course of a year per patient, the MRIs are 85% cheaper than using an endomyocardial biopsy technique. It's just so much cheaper to do a non-gadolinium-based CMR study. And even if you take into consideration the biopsies that needed to be performed in that handful of patients where the clinician just didn't believe the CMR, still, it's so much cheaper to perform an MRI as your initial screening test. So where to from here? We're going to now perform a very similar study in a multi-center way, which will include the pediatric population. We're going to randomize 42 patients in each arm, and we're going to add other non-invasive techniques, such as genomic analysis with GEP and cell-free DNA techniques, as well as other normal biomarkers, troponins, N-terminal pro-BNPs, etc. So we've got this study approved. We've got this study mostly funded, but we're short. We're short, and that's why we're here asking for more. The study is quite comprehensive within the adults in this randomized component. And we're going to add children into this randomized component in the future. But before we do that, we need to actually establish that in small hearts, we can do exactly what we do in the big hearts. So our primary aim is to assess the feasibility of CMR imaging in non-invasive detection in acute cardiac allograft rejection in pediatric populations. And our secondary aim is to undertake this preliminary analysis of the correlation and the reliability of CMR imaging for this non-invasive detection of ACAR in the population. And we hypothesize that this works. It's worked in the adults really well. And we believe it should. There's no good reason why it shouldn't work in the pediatric population. So we've planned an observational, prospective, cross-sectional study of pediatric orthotropic heart transplant recipients. The screening of study participants over a 2.5-year period, and it will involve corresponding CMRs at 1.5 Tesla, the exact same machine that we use at St Vincent's. They've got the same machine over at Westmead Children's Hospital, and where they're performing heart transplants. And we've already been over, we've installed the WIP software into their programs. We've made sure that it works so that we're all set up and ready to go. The CMR procedures will be performed according to the standard scheduled appointments to undergo, that we have patients undergo routine EMB. There'll be 40 pediatric recruits within 1 year of recruitment, and all the pediatric patients will be recruited at Westmead Children's Hospital. So what are we asking for? We're asking for some salary support for a study coordinator at the Children's Hospital. We've already received in-kind support from St Vincent's, where I work, and from the Victor Chang Cardiac Research Institute, and there's some in-kind support from the Westmead Children's with infrastructure support. And the doctors there are really bright, and they're really hardworking, and they're overworked. And we know that to make this a reality, to be able to translate what we've seen work in adults into children, we really need a study coordinator to arrange these and to coordinate the study at the site. And although the doctors are very well intentioned there and are great researchers and great clinicians, they really need a hand to make it a reality. We've received CMR support from the New South Wales Health Department, who will be funding all the MRIs on the project. So this project will bring together a multidisciplinary team of experts in clinical transplantation medicine, advanced cardiac imaging, cardiovascular and physical sciences and clinical trials. It's a collaboration between 2 hospitals, 3 research institutes and the University of New South Wales. It builds on established productive collaborations between myself, Professor Peter McDonald, our international counterparts in Germany and in London, and in addition, it enables the opportunity to foster new and develop new collaborations between us and the Kids Hospital to investigate the feasibility of MRI in detecting ACAR in heart transplantation. And so ultimately, this grant has the capacity to provide meaningful research that is practice changing to children. So I might just conclude with a final slide, which is a shout out from one pediatric patient whom we've had exposure to and who's given me permission to use this, as you'll see in a second. This is Licia. She's a delightful 10-year-old girl from New Caledonia. And she has a dilated cardiomyopathy, and she was airlifted from New Caledonia urgently to the Children's Hospital at Westmead. It was deemed that she needed VAD support, and because of her size, she was moved. She's only 10, but she's 80 kilos, and so she was moved from the Children's Hospital to our adult facility. She underwent a VAD that should normally be retrieved to France, but this couldn't happen for technical reasons, and so she underwent a heart transplantation. The good story for Licia is that she only needed 1 endomyocardial biopsy the week after transplantation. Every single other screening test for rejection has been CMR, and her heart is going like a rocket, and she's done really well. She's been able to return home to New Caledonia, and so she wanted to give a shout out to everybody. So hopefully you can hear this.
We cannot hear it.
Oh, okay. Let me just try and...
You know, I think we need to move on to questions, actually,
Sure,
if that's okay.
that's fine.
All right, thank you so much. So our first question, asymptomatic acute rejection in children is even less common in children than adults. CMR will still require GA from younger children, likely longer than for biopsy. Will your study be powered to see differences in children, and is the risk of GA justified?
So our study, this study is initially a feasibility study, and it will eventually be powered to determine that. But this is the pilot version. This is the feasibility. This is to get the Kids Hospital off the ground. Not all children require a general anaesthetic for an MRI. The youngest ones do, but those that above 8 years old have been able to successfully go into an MRI scanner, coaxed with their parents. So, for example, Licia is 10. She doesn't look like a 10-year-old, but she's 10. Her mother held her hand throughout the whole procedure. These are free breathing scans. You're in there for 15 minutes and you're out. There's no gadolinium. And she tolerated the studies well. The image quality is really good. In the very young, you will have 15 minutes of a general anesthetic for the MRI. But in the older children, you won't need any anesthetic. You won't need any needles at all. So eventually, for the power part of that question, yes, eventually you will need a study that will power. This is a feasibility, and for children that often just won't need anything, any intervention whatsoever, no cannulas, no general. That's the beauty of MRI.
So the next question, echo screening for rejection is relatively sensitive for infants. Have you compared echo findings with CMR or just EMB?
So we have not yet compared echo with CMR. Echo is relatively sensitive, but it's still imperfect, and it can't really detect myocardial edema in the way that CMR can detect myocardial edema. The reproducibility of CMR is certainly better than the reproducibility of echocardiography when you look at T1 and T2 mapping. We can also do the type of strain techniques that are used in echocardiography with CMR as well. There's a huge amount of data looking at myocardial tissue characterization without the use of gadolinium in transplant and non-transplant populations, and this is quite robust data, perhaps by looking not just at the way that the heart functions, but also at the tissue character, we might be able to detect rejection at an earlier stage rather than when it's caused dysfunction.
And so one very quick last question. What will be the age range for the pediatric cohort?
The age range will be all ages. So they're transplanting children up to 16 years of age over at the Children's Hospital, and the youngest patient will be a year. I think 1 year is the cutoff that we've used in our clinical trial. If their clinician feels that they are safe enough to have a general anaesthetic for an endomyocardial biopsy, then they will also be recruited to this study.
Thank you very much, Dr. Jabbour. We're going to move on to our next presentation. That presentation is entitled Heart Biopsy by Microcatheter Technique and Molecular Biology Analysis for Diagnosis of Heart Transplant Rejection. The finalist is Dr. Emil Najar from the Karolinska Institute, Stockholm, Sweden. And this proposal is presented by Rikard Granqvist. Dr. Granqvist.
Thank you. And thank you to ISHLT and Enduring Hearts for this opportunity to present our research project. My name is Rikard Granqvist, and I'm representing Karolinska Institutet and presenting on behalf of Emil Najar, also Karolinska Institutet and Karolinska University Hospital, where he is the VAD director. Our research concerns endomyocardial biopsy, and I will give some extra thanks to Professor Jabbour for providing some important background on the use of endomyocardial biopsy and its limitations. EMB is used routinely as described to surveil transplanted hearts for acute rejection, and typically with a high frequency in the immediate term after transplantation, with decreasing frequency during the first year when risk of rejection is the highest. It is also performed for cause when there is clinical suspicion of rejection outside of the biopsy schedule. It is considered a gold standard, and although non-invasive blood testing and indeed cardiac magnetic resonance has gathered interest in recent years, a positive non-invasive test is usually confirmed by invasive diagnosis using biopsy and histology. EMB and histology has limitations, however, as mentioned. First, regarding the procedure itself, EMB was invented in the 1950s, and there has been quite limited innovation in how the device looks and functions, and the technical approach since then. The bioptome features a forceps-like design, which is quite rigid and large compared to many other endovascular tools, which is required for it to function. Usually access is gained via the jugular vein to get a straight path to the right ventricular septum, where samples are taken for safety reasons. If the free wall is sampled, there is a risk of tamponade, which can be fatal. And when repeat biopsies are performed, there is an increase in scarring of the septum wall tissue over time, which can make it difficult to acquire a representative sample of myocardial tissue for histological diagnosis. Repeated traversal of the tricuspid valve is also thought to increase the risk of significant regurgitation. To combat these limitations, we have developed a novel endovascular device for EMB, which is significantly smaller. Here is a standard 7 French device. Here is the smallest clinical pediatric device, and here is our device. And it also features a different sampling mechanism, which creates a much smaller impression in the ventricular wall, decreasing the risk of tamponade and local thromboembolism formation. We have tested the device. We have tested this device in animal trials in swine in a clinical grade cath lab, just as you would in clinical practice, and found no complications. in over 30 animals and hundreds of biopsies performed, including a longitudinal follow-up study where biopsies were performed multiple times during a 2-week period to look for complications that were not immediate, and none such were found. We also performed a comparative study of EMB with a clinical device and the micro EMB device in a high-risk scenario where we performed a large number of right ventricular biopsies and also sampled the free wall, not just the septum, to simulate a situation where complications could occur. And here we found a significant and quite large difference in short-term mortality due to tamponade. The microbiopsy device has a lot of benefits with regards to safety and sampling flexibility, but it has a major drawback, which is that samples are too small to perform traditional histology on. And this brings us to the limitations with histology for rejection reactions. It has been demonstrated several times, including in the cargo studies, that there is a large inter-rater variability among pathologists and between centers with regards to rejection grading, especially in mild to moderate, or rather the cutoff between moderate and severe rejection reactions. To improve the diagnostic accuracy of EMB, there has been a development of molecular assays, typically based on gene expression tests. The most used test is a molecular microscope, referred to as MMDX, and by Halloran and colleagues, which is now marketed by One Lambda. It is based on kidney rejection originally, where a large number of rejection-associated transcripts were identified and then carried over into heart. And this technique can more reliably distinguish between different grades of T-cell mediated reaction as well as antibody mediated rejection. However, this technique could possibly also be improved, and this is where RNA sequencing comes into the picture. We believe that RNA sequencing and other modern molecular analysis methods of gene expression may increase the sensitivity and specificity of molecular diagnosis in rejection by being able to detect a larger number of transcripts in the tissue, including different types of RNA, not only mRNA and but long non-coding RNAs and microRNAs, which could contribute to the phenotypic picture which describes rejection and perhaps can have prognostic implications with regards to rejection. This is just an example of the use of RNA sequencing on EMB, where a large number of around 1,000 transcripts were found to be associated with heart failure with preserved ejection fraction compared to those with reduced ejection fraction. In our project, we are aiming to verify that the microbiopsy technique is possible to use with current molecular assay methods, specifically MMDX, but also microarray in general and RNA sequencing on human tissue, since all studies up to this point has been performed in swine. We will also explore the development of novel assays based on RNA sequencing in patients surveilled after transplant. So in study 1, we are acquiring samples with the microbiopsy device from explanted hearts and from tissue harvested from non-failing native hearts, that is, patients undergoing CABG, where samples are or tissue is harvested intraoperatively, and then samples are taken from the tissue. We then perform microarray, MMDX, and RNA sequencing on these samples, and compare the quality of samples to reference tissue. Compare the quality of the RNA yielded from microbiopsy to reference tissue from the same patients. In study 2, we are performing RNA sequencing on EMB samples, just conventional standard EMB samples gathered during routine surveillance and for course biopsies at the transplant center in Sweden, in Gothenburg. And we then perform RNA-seq and compare expression profiles gathered from RNA-seq with the rejection grade on histology, as well as clinical follow-up data such as graft dysfunction and changes to immunosuppression regimen, as well as all-cause mortality. We follow patients at least 3 months after biopsy, but we follow patients for the entirety of the study. And in summary, this study is in summary, this study is hopefully going to benefit patients by providing a less invasive and safer technique for endomyocardial biopsy, which may have fewer complications, both with regards to access site, since the device is smaller, it requires a smaller access site, and also with regards to complications in the allograft. We also hope to improve the diagnostic capability of EMB by using molecular methods, and by using microbiopsy we may decrease the risk of non-usable samples of scar tissue. Down the line, we think we may improve the use, may find use for EMB and molecular methods in the pre-transplantation diagnosis and management of patients with new onset heart failure before they need to get their transplant. Thank you.
Okay, thank you so much, Dr. Granqvist. We have some questions for you. First question, is the yield of the microbiopsy technique sufficient for histologic analysis? Do you need more samples to obtain the same data?
Yes, well, the yield is not, in our opinion, enough for histologic analysis, which is, I mean, you typically take 5 traditional EMB samples, which would represent a large amount of micro EMB samples when it comes to tissue size. So micro EMB is designed as a molecular only or molecular first technique. So moving it to clinical practice requires that molecular testing will be the primary technique, and then perhaps in equivocal cases where the diagnosis is still unsure, traditional EEB might need to be supplemented. But hopefully, if we combine non-invasive testing with microbiopsy, then that could be sufficient in many cases.
Thank you. And I'd like to remind everyone that if you have questions, please put them in the Q&A, not the chat. Next question. Are all your proposed samples from adults? Biopsy mRNA assessment has not been clinically validated as a sole predictive tool in adults or children. Do you think your study will allow any conclusions beyond feasibility of running the test, since you will not have comparison regular biopsy samples?
Well, we will only in this study show the feasibility of using micro EMB for molecular diagnosis, to see basically if MMDX and microarray works as well. For us, it has worked very well for us in swine, both microarray and RNA sequencing. But just as you say, molecular diagnosis is not validated yet as the only technique in adults or children. These samples will be taken from adults only, because for practical reasons and our ethical grant support at the moment, but we will obviously extend this to the pediatric population when the feasibility has been established.
So just to clarify, for this proposal, will there be any pediatric patients enrolled? Is the device approved for use in pediatric patients?
The device is not yet approved for use in humans at all, since it's at the research stage. There will be no pediatric patient samples included in the study. No.
Okay, thank you very much. And we will move on to our final presentation. And that presentation is entitled Analysis of Associations of Age-Dependent Patterns of Cardiac Allograft Vasculopathy in Pediatric Heart Transplanted Patients with Modifiable Risk Factors. And this proposal is from Madeleine Orban, University Hospital of Munich, Ludwig Maximilians University, Munich, Germany. Thank you.
Thank you. Thank you for the opportunity to present our research project. It's a great pleasure to conclude a day full of fascinating proposals. I hope to get you just as excited about ours. So let me introduce you to our research project. Using intracoronary imaging, we aim to quantify patterns of cardiac allograft vasculopathy in pediatric patients and assess whether there are associations with modifiable risk factors. So I'm presenting this research proposal on behalf of my colleague, Sarah Ulrich from the Department of Pediatric Cardiology and myself from the Department of Cardiology. We share a long interdisciplinary research cooperation now and could show that age plays a relevant role in pediatric and adult transplanted patients regarding cardiac allograft vasculopathy, CAV. This was the case regarding the prevalence of absolute and relative intimal hyperplasia in optical coherence tomography, and also age played a role regarding the associations of risk factors regarding absolute intimal hyperplasia in optical coherence tomography as well. The relevance of age in cardiac allograft vasculopathy has been shown previously in large registry studies that mainly focused on CHV defined as angiographic changes mainly. And here you can see that age plays a role regarding the prevalence of CAV. Yet, independently of the age cohort, CAV remains a major limiting factor for survival. So it seems most important to better understand what is CAV and the different age cohorts, what are the differences, how can we improve prevention and treatment strategies. To improve these treatment and prevention strategies, we have to understand what CAV is an individual patient. As CAV is a complex disease that has been shown previously, and the most frequently described pattern of CAV is intimal hyperplasia. Yet it is important to note that pathological studies have reported that beyond intimal hyperplasia, there are other different morphological manifestations, and specifically atherosclerotic plaques. And interestingly, this was not only the case in adult patients, but also in pediatric patients. And based on those findings, they also raised the question that it seemed that it was not only related to the donor age, but didn't have enough data to go on further. This high prevalence of atherosclerotic plaques after transplantation has been confirmed also in intracoronary imaging studies, and particularly in adults. In pediatric patients, there's less data, and while this seems very important, yes, there's a lack of studies. In adults also, it has been shown that there's an association of those plaques with angiographic manifest CAF that is known to be prognostically relevant. And it was associated with percutaneous coronary intervention, progress of cardiac allograft vasculopathy, and also stenosis at the vessel level. This leaves us with the very important questions of, we know that in pathological studies, there have been some atherosclerotic lesions that have been described, but are there differences between pediatric and adult transplanted patients? Are those lesions as clinically relevant in pediatric patients, or is there an age dependency? And finally, are there some age-dependent risk factors that could potentially be targeted in selected pediatric patients that could be at risk? So with these questions in mind, we chose to use the findings we see in optical coherence tomography. And why did we choose OCT? OCT is an intracoronary imaging tool that we use in routine post-transplant follow-up at our center in both pediatric and adult transplanted patients. We do that in line with the German guidelines for pediatric cardiology and also with the recommendations of the European Association of Cardiovascular Imaging for post-transplant care. OCT represents a valid tool for in vivo plaque assessment, as previous studies have shown that when comparing OCT findings performed in hearts after autopsy and histopathological findings, there's a high sensitivity and specificity. Other studies later showed also there's a high inter and intra-observer reliability, which is of course important. And finally, there's a consensus on how to define those plaques found in OCT that also has been recently updated. We chose OCT over IVUS because of its higher spatial resolution, which is of course important, particularly in pediatric patients. And it also provides a better plaque characterization than IVUS. Importantly, it keeps a good correlation with IVUS for the measurements of maximal intimal thickness that currently defines, or it's the parameter that is currently used to define intimal hyperplasia, and which is the endpoint in studies assessing immunosuppressive medication. Let me show you some examples from OCT findings and why we came to this project. In the middle, you can see a normal vessel wall. Intima layer is very thin. When you compare to the findings in the upper part of the slide, you can see on the left side, there's an eccentric intimal hyperplasia. And on the right side, it's a concentric intimal hyperplasia. But there's not the only findings we have in OCT. Quite commonly, we find fibrotic plaque. And this is not the case only in adults, but also in pediatric patients. In adult patients, we also find very commonly other types of plaque. We find lipid and calcified plaque, which we believe is less common in our cohort of pediatric patients, but let's find out. And how do we want to find out concretely? We want to include 40 pediatric patients and 70 adult patients and assess the patterns of cardiac allograft vasculopathy we find in optical coherence tomography. We want to assess both plaque and of course intima, as this is the baseline, the standard. For the plaques, we want to compare whether there's a difference between the prevalence of the different plaque morphologies, fibrotic, lipid and calcified or mixed types. We want to compare between those cohorts. We also want to quantify the plaque burden, where we'll provide measurements that we perform at every millimeter of each plaque and then see how is the plaque burden in the analyzed vessel. And of course, we will also perform reliability analysis. For the intima, we will provide data regarding absolute and relative intimal hyperplasia, with relative intimal hyperplasia being emerging concept where it's defined by the intima to media ratio being superior to 1. For all these findings, we will assess whether there is an age dependency. In addition to comparing both cohorts, we also want to use the continuous variables of recipient age. And also when you talk about age after transplantation, of course, you have also to take into account donor age. Donor age has been shown to impact on cardiac allograft vasculopathy to some extent, probably mainly early after transplantation, but this is not fully understood yet. Then we will evaluate the clinical relevance of our findings in the pediatric patients and compare with adults. We will see if there's an association with angiographic CAV, which we will define as overall CAV based on the ISHLT CAV classification, and also evaluate if there's an association with stenosis at the vessel level. And then later on also with CAV-related complications, such as maybe a worsening of left ventricular ejection fraction. Finally, we will assess if there is an age dependency of associations with risk factors. Here, we want to mainly focus on potentially modifiable risk factors and the corresponding laboratory findings. So why is that? At first glance, this might seem surprising, maybe not if you had the first presentation, but there's a high prevalence of cardiovascular risk factors, both in pediatric and adult transplanted patients. The reason why that is probably that these cardiovascular risk factors seem to be related to the immunosuppression itself as side effects. So I brought together for you the description of the frequencies of, just as an example, arterial hypertension, dyslipidemia, and hyperglycemia that you find in the official product characteristics. And you can see there's a high frequency. Importantly, those risk factors, particularly arterial hypertension and dyslipidemia, have been related to angiographic cuff and also to intimal hyperplasia. And finally, there's this emerging concept, which we find quite interesting, of insulin resistance syndrome that had also been related to angiographic cardiac allograft vasculopathy, and where we could show in adults that there's an association with fibrotic plaque burden. This insulin resistance syndrome, well, not the syndrome, but insulin resistance is associated also with immunosuppressant medication. This has been shown in previous studies, and it has also been shown quite importantly that it's potentially reversible. So this might be important. And finally, it clusters the risk factors such as arterial hypertension, dyslipidemia, and hyperglycemia. So this might maybe link the risk factors to the findings we have, but we will see. So with these findings, with this information, we hope to provide some pieces to a larger puzzle for a better understanding of what can be cardiac allograft vasculopathy after transplantation, and particularly in pediatric transplanted patients, and maybe some targets for primary and secondary prevention in the future. So thank you very much.
Thank you, Dr. Orban. We are open for a couple of questions. I know we're a little bit over time. I'll ask just a quick question while we're waiting for the Q&A to populate. What's the age range for your 40 pediatric patients and projected age range?
Yes, the youngest would be around 4, but the median age would be approximately 10 to 12, I'd say.
Okay, thank you. So we have first question. Is your pediatric cohort big enough for conclusions given the heterogeneity of patients, age, time post-transplant, underlying disorders, therapies?
That's a very good question. And we would see it's a cross-sectional study, and we see it more as hypothesis generating, with the hope that we can, based on that, maybe, if possible, with other cooperation, confirm, or in whatever direction that goes, then see what we find in larger cohorts. But we have to start somewhere, and this would be the start.
Okay, thank you. Next question. Early detection is a desired characteristic of diagnostics for CAV. What is the earliest manifestation of CAV detected by OCT, and what would be the youngest? Oh, we already answered that. What would be the youngest age? You said 4 years. So just the first question.
I'm sorry. So I didn't get it completely. So the earliest detection.
When the earliest manifestation of CAV detected by OCT.
The earliest manifestation we find based on intimal hyperplasia, you mean?
Yes.
Oh, I don't have, I couldn't answer that. I would have to check in the data.
Okay. All right. I see no additional questions. So I would like to thank all presenters. Wonderful, wonderful presentations today. So we thank you for your time and all of the hard work you've put in submitting proposals and presenting them today. And so this concludes our session. Thank you so much, everyone, and have a great day.
Thank you.
Bye-bye now.
Thank you.
Thank you very much.
You're welcome.
Thank you. Have a good day.